So we're still on the subject now of talking of the cytoskeleton and the various proteins involved with the cytoskeleton structure. So we just recently talked about actin and how that's a major protein that makes up the cytoskeleton. And another thing is that the actin also has a role in the physiology, or has a physiological role as well in terms of contributing to muscle contraction. Another filament that we're going to look at now is called the microtubule. Now the microtubule also has various roles within the cell. One of its main roles is to serve, again, a structural role, so providing some shape and stability to the cell. And it's also going to act as a track system. So that is where motor proteins can walk along microtubule tracks, and they do this by carrying vesicles or helping to carry vesicles and organelles around this within the cell. So remember, the vesicles that are moving, say, from the ER to the Golgi, or from the Golgi back to the ER, or from the trans-Golgi network to the plasma membrane, they're not floating around. They are being carried by motor proteins that walk along these various tracks. And these tracks are made up of both actin and now this other system called the microtubule. So we've talked about actin. We've seen how actin gets made and polymerized with G-actin, individual G-actin monomers coming together, building a filament that was then polarized with a plus end and a minus end. And these G-actin monomers were activated with ATP, and then over time the ATP hydrolyzed to ADP, and then the actin filament was subject to treadmilling or falling apart. Well, the same similar storyline is true for the microtubule. The microtubule also consists of a building block, but this building block is not an individual monomer like the G-actin was, but it is a building block consisting of a dimer. So two monomers, or two proteins, bound together, forming what's known as an alpha-beta tubulin, or an alpha-beta tubulin dimer. This alpha-beta tubulin is the building block for a microtubule. So this structure of the alpha-beta tubulin is illustrated above. Now, it's an alpha and beta subunit, so there's two monomers joined together that form the dimer. I'm going to point out some key features of this building block, the alpha-beta tubulin, and then I'll go in and describing how then this alpha-beta tubulin, groups of them come together in order to establish the actual microtubule itself. So first, let's look at the alpha subunit. The alpha subunit, as you can see, or hopefully you can see, is composed of, as you know, beta sheets, alpha helices. It's kind of a globular-like structure. It also contains a nucleotide binding site. So now instead of ATP like we saw with actin, what we're going to see with microtubules is GTP. So guanine or guanosine triphosphate rather than adenosine triphosphate. Now in the alpha subunit, there is a GTP that is bound to it. But this GTP in the alpha does not serve as an energy, but serves more of a structural role. So a GTP will always be bound to the alpha subunit of the alpha-beta tubulin. Then the other part then is the beta subunit. Now the beta subunit looks similar to the alpha, composed of alpha helices, beta sheets, also globular-like, but it has also a GTP binding site. Okay. Now, when GTP binds to the beta of the alpha-beta tubulin, this GTP in the beta is going to provide energy and signal the alpha-beta tubulin to go ahead and hook up with other tubulins that also have GTP in their betas and be added to the plus end of a pre-existing microtubule, or for other alpha-beta tubulins that have GTP bound to their beta to come together and start to form a new microtubule. So basically what I'm trying to say is that the GTP in the beta tubulin, or in the beta subunit of the alpha beta tubulin, is what's needed to start polymerizing or assembling a microtubule. The GTP in the alpha is simply a structural significance. Okay? So when we go through these episodes now talking about microtubules and we talk about beta tubulin, I'm referring to the fact that, or when I say there's a GTP beta tubulin, I'm referring to the fact that there's a GTP bound to the beta subunit of the alpha beta tubulin dimer that is then subject to being added to a microtubule at the plus end. Okay? And that's how it's normally described when you're discussing the alpha beta dimer. No, you don't usually hear alpha beta tubulin dimer coming together. Some people just simply say GTP beta tubulin comes in, hooks up to the plus end of the microtubule. Because again, the GTP in the alpha we don't care about because that's always there. But it's whether or not you have GTP or GDP bound to the beta tubulin that determines if it's the plus end or the minus end of the microtubule. Again, as we discuss this and go through these episodes, you'll see what I'm trying to say. So now continuing on then with the assembly or the polymerization then of a microtubule. So again, once you have now a group of these beta tubulins or alpha beta tubulins with GTP bound to the beta, they're going to come together and they will form a string of alpha beta tubulins. This initial string of alpha beta tubulins with GTP in the beta is called a protofilament. Okay? Then what happens is that a whole bunch of these protofilaments get made and established into a flat sheet. Okay? So similar to a flat sheet of paper. So each individual protofilament would line up and form a sheet, a flat sheet. Then this flat sheet of protofilaments, which again are composed of alpha beta tubulins, eventually roll up. Again, just like a piece of paper, they roll up into a hollow tube, and this hollow tube-like structure then is the microtubule. Okay? And then as you can see then also, this microtubule is polarized. So it has a plus end and it has a minus end. Again, this has nothing to do with charges. The plus end is where beta tubulin with GTP bound gets added, so where we see growth happens at the plus end, and at the minus end we will find an accumulation of GDP beta tubulin. So that is GDP in the beta tubulin of the alpha beta dimer, and that accumulates at the minus end. And just like with actin, when actin had G-actins with ADP, those G-actins were subject to being taken off by cofilin or falling off. The same thing goes with the microtubule. GDP beta tubulins are subject to being taken off by disassembling proteins, which we'll talk about, or falling off. So this also clues you in to the fact that the beta subunit of the alpha beta tubulin dimer, the beta has a GTPase activity. So when GTP comes in and binds to the beta tubulin of the alpha beta dimer, the alpha beta goes ahead and adds to the plus end of the microtubule. But then over time, that GTP that was in the beta subunit eventually gets hydrolyzed into GDP. Okay, and that's only at the beta tubulin. Remember, the GTP in the alpha is always going to stay GTP. But in the beta subunit, the GTP gets hydrolyzed to GDP. And again, we find an accumulation of GDP beta tubulins at towards the minus end. And then again, these are subject to coming off. And then now what we're going to be looking at then is the roles that the microtubule plays.